Hey everybody, we're the Uncalled Four, and today we're going to be talking about how companies like Ancestry.com and 23andMe are able to determine someone's ethnicity just from their saliva. So, a good place to start would be to define what ancestry testing actually is. When a person conducts ancestry testing, they look at predetermined segments of a genome and compare these segments to reference groups in order to obtain an estimation of your ethnic background. The largest two ancestral companies are Ancestry DNA and 23andMe, with 16 million and 10 million people in their respective databases.
This type of ancestry DNA testing is also known as biogeographical analysis, or BGA, where an estimation of one's biological or ethnic background can be determined based on a DNA analysis. In this test, an individual's DNA is compared to reference sets, also known as ancestry informative markers, or AIM. There are three different types of DNA testing. Y-DNA testing looks at the Y chromosome. However, it is limited to men as it requires the Y chromosome, and it is more subject to mutation in comparison to methylated mTNA, which makes its results a little more volatile. mtDNA testing looks at mitochondrial DNA. However, it's limited to tracing the matrilineal ancestry since the mitochondria are passed down from the mother. Autosomal testing, which is the most common type of testing used for biogeographical analysis or ancestry testing, can be applied without limitation to sex and can trace both the mother's and father's side.
Here's a general overview of how autosomal testing works. Autosomal testing is the most common test for ethnicity estimation. Once DNA is obtained, which is usually through saliva, DNA will get amplified or make copies, normally through PCR. Next, DNA is placed on an Illumina chip or similar microarray to compare SNPs, and then PCA is employed to reduce noise in the data. Now we'll go on to each term and process. PCR. Polymerase chain reaction is the common lab technique used to make copies of a specific DNA segment. PCR has three general steps. First, heat to about 96 Celsius to separate DNA double strand. Second, cool to about 55 to 65 Celsius to allow the primer, which is designed based on what segment of DNA want to be copied, attached to the template DNA. Third, heat to about 72 Celsius. Uses the DNA polymerase named Taq polymerase to extend the primer to construct a new DNA strand. This cycle is repeated to get more copies. Next, hand it to Steven.
Next, we'll talk about the sequencing chip used by the two biggest ancestry companies, the Illumina chip. This chip has thousands of beads assembled onto the chip's optical fibers. Each bead contains a short sequence of nucleotides that find and mark the SNPs of a person's saliva DNA. For those who don't know, an SNP is a single nucleotide polymorphism, or mutation, which are the places where DNA mutates most frequently and where DNA most frequently differs between people. So, some pros of the Illumina chip are its extremely high accuracy in comparison to other sequencing chips, its low cost per unit of data, coming in at about 50 cents per megabyte of data, which is extremely low in comparison to other chips. It has a high throughput, which means that tons and tons of DNA molecules can be sequenced at the same time. But some cons, as a result of this high accuracy, it does have a long sequencing runtime at 27 hours per chip. It has shorter read lengths than other chips, but shorter read lengths do give you higher accuracy. And lastly, it has an extremely high and, for most people, unfathomable machine cost at $125,000.
Once data has been gathered from the microarray, it needs to be simplified in order to visualize it. To do so, principal component analysis, or PCA, can be employed. PCA reduces noise in the data without losing the significance behind it. Some general terms to know: Variance at a high level just means a measurement of the distance between two or more values, and the principal component is a vector that runs through the data where there is most variance and acts similar to a best fit line. In DNA ancestry testing, the data points are the SNPs identified by the microarray. Because there are thousands and thousands of SNPs that are analyzed, this results in a dataset with too many dimensions to properly analyze it. The goal of PCA is to take this dataset and reduce it down to 2 or 3 dimensions, making it much easier to process. To do so, 2 or 3 principal components are identified and serve as the new axes for the data points. This results in a dataset that can be visualized in a 2D or 3D environment. This makes it easy for it to compare to reference sets and estimate the individual's genetic makeup. By reducing the number of dimensions of the dataset, it makes it much easier to process and analyze the data without losing the significance. To summarize, DNA ancestry tests use autosomal testing, which identify SNPs in an autosomal or X chromosome. SNPs, or single nucleotide polymorphisms, are locations where variations in single nucleotides are common. A common type of microarray used in DNA ancestry testing are the aluminum microarray chips, which hold thousands of beads, each binding to a specific SNP site. To process this raw data, PCA is employed to reduce the dimensions in the dataset so it can be viewed in a 2D or 3D environment. Once the data has been processed through PCA, it can then be compared to reference groups to gain an estimate of the individual's genetic makeup.
Now that we've given a more general overview of ancestry testing, we'll do a brief comparison between the 2 most prominent names in the ancestry testing industry, Ancestry DNA and 23andMe. A major difference is that Ancestry DNA only tests autosomal DNA, which, to review, is a type of testing that looks at autosomal chromosomes and can be used without limitation to sex. 23andMe also uses Y-DNA and mtDNA, looking at Y chromosomes and mitochondrial DNA for a more extensive analysis. Notice that Ancestry DNA's autosomal SNPs are almost twice the size of 23andMe's, lying at 28,000 versus 16,000, which could be potentially due to 23andMe's more widespread look at ancestry. In addition, Ancestry DNA has around 16 million people in its database, while 23andMe has 10 million. The differences between these 2 companies are relatively minimal, but the results they yield can be quite different. Ancestry testing's pros and cons. One of the main advantages is that it can give an estimate of heritage for those who are curious about their general origins. Its analysis by region can allow people to discover possible roots that they never would have known previously. This could be particularly beneficial for those who are adopted, whose histories are more unclear. As a result, ancestry testing has great potential in helping people connect and reunite with long-lost family members. Unfortunately, the truth behind ancestry testing is that the tests are simply a guess. Your individualized results are based on comparisons to the database of that particular company and are only a prediction of where they think you could be from. While accuracy is pretty high when it comes to reading each of these markers, presentation of data in the typical percentages or pie chart format suggests results are definite. This is misleading and could even reinvigorate age-old racial stereotypes by creating a sort of divide where people are led to believe that those of different races are biologically or essentially different. Other limitations to ancestry testing are that the reference populations vary across companies. Because different companies are comparing your data to their own database of samples, the exact results you get will change between companies. As they get more samples and reclassify, your percentages could shift likewise. Not only does this mean that small percentages are often unreliable, but also adds to the variability of your results. Another downside is that the ethics and minimal confidentiality behind the business mean that the companies are in full control of your DNA and private information. Once you send in your tube of saliva for testing, companies could sell it and use it in ways completely out of your control.
Alright everyone, this is the end of our presentation. Thanks for tuning in and for the amazing 3 weeks we had. Special thanks to Philip, Josh, the TAs, Marcus for teaching us when Kunal wasn't around, and of course, Kunal for helping us through this comp bio struggles, and for being the best SalCon we could ask for. Thanks, guys.